Hello, and welcome to JAMA Evidence, our podcast series based on core issues in evidence-based medicine. Today, we will be discussing Genome-Wide association studies. I'm your host, Dr. Roger Lewis, Senior Statistical Editor for JAMA and co-editor of the JAMA Guide to Statistics and Methods series. I'm here with Professor Jerry Rotter. Dr. Rotter, thank you so much for joining us today. Can you go ahead and tell our listeners a little bit about yourself?
Yes. I am currently director of the Institute for Translational Genomics and Population Sciences based at the Lundquist Institute at Harvard-UCLA Medical Center, the southern campus of the UCLA School of Medicine. And I'm also a professor of pediatrics and human genetics at UCLA. My long-term research interest over 4 decades has been exploring the genetic basis of common chronic diseases, especially cardiometabolic diseases, such as coronary artery disease and diabetes, but also blood pressure, lipids, adiposity, and other related conditions.
Thank you so much for joining us today. So in your article, you're talking about genome-wide association studies, an approach to evaluating diseases that may have very complicated relationships with the underlying genetics of affected persons. But first, let's start with the initial situation that you mentioned, which is a Mendelian genetic disease. Go ahead and tell us what that is and why the diseases you're targeting are so different.
So Mendelian genetic diseases are diseases due to the change in a base pair. That single change in a base pair of the DNA code results in a phenotype, meaning a description. Good examples are Marfan syndrome, sickle cell anemia, Huntington's chorea. These are all conditions where a change in a single base pair in the genetic code leads to a medically relevant disorder. Another good example would be cystic fibrosis.
So, Jerry, the diseases you're studying are not that simple. So go ahead and explain to our listeners what it is that makes them so complicated with respect to the genetic influence on the disease.
So it turns out, as opposed to the Mendelian conditions that we just referred to, most common chronic diseases that the internist will see, such as chronic lung disease, coronary artery disease, type 2 diabetes, are due to the effect of literally hundreds of variants in genes acting simultaneously to provide risk and eventually a disease pathogenesis. So it's not the effect of one gene at a time, it's a whole concert of genetic variations acting together that provide the susceptibility. And the term for that is polygenic disease, i.e. many individual genetic contributions.
So in studying these complex relationships between many genes and the occurrence or the manifestation of disease, it must be hard to have a specific hypothesis when you start out. So how is it that the genome-wide association approach can proceed if we don't really know exactly what we're looking for?
That's a superb question, and it's precisely because we don't know what is the cause that the genome-wide association study works. It turns out of the 30,000 known coding genes that code proteins, we don't know actually what most of them do, and we don't know their relationship to disease. And so much of human biological research is pretty much working in the dark with some brilliant insights periodically. What a genome-wide association says, I don't know what causes the disease, but I will let the fact that the genes in the human genome are not random pieces of DNA in the nucleus, but are like beads on a string of the 23 human chromosomes. And I can use their location to find genetic influences. So the fact that genes are organized in structures on chromosomes is the tool that we use to identify the genetic variants that actually predispose to these various different conditions.
So in working to identify the locations of the genes that influence the disease, and I think you call these effector genes in your article, we're taking advantage of other things we know about the locations of genetic variability on the human chromosome, and specifically the so-called Snips. So can you talk about what a Snip is and what it is we know about Snips that allows us to use them as tools to locate other genes, even when we don't know what it is we're looking for?
Yes. So it turns out human beings are extremely variable in their genome. The genome is the collection of the 23 human chromosomes, which is about 3 billion base pairs. Base pairs are the DNA letters that make up our genetic code and make up the chromosomes. So it turns out approximately every 100 base pairs, there are variations. So just think about it a second. If there are 3 billion base pairs and every 100 base pairs there's a variation, that means your DNA has a variation 30 million times. So that's an enormous amount of variation, and that varies between you and your neighbor, between you and your classmates, and to some extent between you and your family members. And we can use that variation by comparing the variation in one person versus another at the same time, whether that one person or another has a specific disease, such as type 2 diabetes, and ask the question, is the variation along the chromosomes related, meaning associated, with the disease of interest?
So one leverages these so-called SNPs to do that. What is a SNP, and how does it help you identify the locations of genes that you are looking for?
So I remind you that the DNA alphabet has got 4 letters, A, G, C, T, and a SNP is, as you go along the gene, it's a variation in one of those letters on one chromosome versus the other chromosome, or the same chromosome in one person versus another. So they're called single because it's a change in one base pair. They're nucleotide because the genetic code is made out of nucleotides, the 4 AGCT. And they're a polymorphism because it means they vary. If they don't vary, they're what's called monomorphic. So we take advantage of the variation along the genome, which is termed polymorphism, a fancy term which just means it varies.
So if all of us have these frequent variations in these particular base pairs that constitute these SNPs, how do we use those at a genome-wide association study to locate regions of the chromosome that are then associated with the target disease?
So very simply, let's take a simple example. Person A with diabetes has a SNP. We'll call it variation. At that SNP, he has a letter A on one chromosome, and in fact, maybe even a letter A on the second chromosome. We go to his non-diabetic neighbor. That person has an A at the same SNP, at that same location, but on the other chromosome has a B. So the diabetic has 2 A's. The non-diabetic has an A and a B. And so we simply count all the A's in the diabetics versus all the B's. And for those regions where there's a difference in the A's and B's in a diabetic versus a non-diabetic, that region is associated with type 2 diabetes. And we do that along the genome in a typical genome-wide association 1,000,000 times, starting with chromosome 1 all the way down through the sex chromosomes X and Y.
So if we're looking at a large number of different locations of genetic variability to assess their association with the disease, how do we get enough data to actually be able to sort this out?
That's an excellent question. So what we need to do is we have to have an adequate study size, 1,000 diabetics, 1,000 controls. The initial genome-wide association studies had a couple of thousand cases, a couple thousand controls. We are now up to studies. For example, we recently published a paper in Nature on the largest genome-wide association study of type 2 diabetes that was done in 5 ethnic groups and included 2.5 million people. So if you have a couple thousand up to a couple million people, you have enough data for each genetic SNP along the genome to see if there's a difference between the cases, in this case type 2 diabetes, and the controls, in this case the non-diabetics. And we actually found now basically 1,289 different SNPs along the genome in 611 loci. Loci are the addresses to which the SNPs reside. So there are at least 611 regions along the 23 human chromosomes that contribute to type 2 diabetes in the population.
So if you have such a large population and you're looking at so many different locations, it seems like there would be lots of ways you might have false positive results, things that pop up that just reflect noise as opposed to a real association. How do you make sure you don't get fooled by false positive results?
That's a superb question because, of course, it goes back to the old problem of multiple comparisons and statistics. I remind the listener that in conventional studies, we use a p-value of 0.05, which means that we accept that there may be not a true association 1 out of 20 times, even though we see that level of significance. The problem that we're having with a genome-wide association study is we're not doing one test, we're doing a million tests. So the way that the statistical field has dealt with that in a straightforward way is to adjust the level of significance and make it much, much more rigorous. And so instead of accepting a p-value of 0.05 for any given association, we make it actually 1 millionth that of. So for a variation in a Snip to be considered significant, it must meet a p-value of 5 times 10 to the minus 8th, which is roughly 0.05 divided by a million.
As I understand from your article, this approach takes advantage of the fact that genes or other variations in the genome that are physically close to each other tend to get inherited with each other. So there's a correlation that's based on how close one area of genetic variation is with another area of genetic variation. What is that called, and how does that help you use a SNP to locate the gene you're really interested in?
So that phenomena is called linkage disequilibrium. It comes from the term, first of all, linkage, which is when two genes are on a chromosome and can be inherited together in families. And that's how we actually did initially genetic studies using genetic markers in families. But it turns out that the human species is a relatively new species. And so there are whole chunks of our DNA that are inherited as a unit, not only in families, but across the population. So that SNP A in location 1 and SNP D in location 2 may often be inherited together in the population. And these chunks of DNA, that phenomena is called linkage disequilibrium because they're not in equilibrium. When you know something about the SNPs in location 1, you actually know something about the SNPs in location 2. And if they were in equilibrium, you wouldn't. And because of that phenomena, while that helps in the beginning of GWAS studies identify the genetic region, it also causes then problems later on because then we don't know which SNP right away in the region is the SNP that is responsible for the genetic association.
So you might identify multiple SNPs that are associated with a disease. How do you sort out which is the one you're most interested in?
So we do a series of things. One is we do what's called a conditional analysis, where we look at each SNP, conditioning it on the other ones, and see which ones, shall we say, remains when standing. Now that helps a lot, but doesn't resolve it completely. Another phenomenon that we take advantage of is the linkage disequilibrium patterns turns out to be different to some often significant ways between one ethnic group and another. So if you do your GWAS studies in the major ethnic groups around the world, that can help again narrow the region for your trying to find the causative SNP. And then finally, there are new tools where you compare the SNP that you find with what's called transcriptomics or expression of nearby genes. You ask if the SNP that you found that was associated with diabetes, is it associated with the level of RNA expressed in a nearby gene? So those different methods now can help narrow in a number of cases and get you to what we think is the actual causative variation or causative SNP.
So in these studies, when they're conducted, you identify a set of SNPs that are associated with the disease in question, and you identify loci where those SNPs are located. What actually are those SNPs? Are they directly related to the disease? Are they just markers of locations? Or how is it that they become associated with the disease in question?
Well, there are basically three explanations. In a small percentage of the cases, 5 to 10%, we've learned that the SNP is a variation in the nearby coding gene or a somewhat nearby coding gene. And that SNP is a variation in the gene that affects the gene's function. But that's the minority of cases. When we first observe these. In the majority of cases, we find that the SNPs that are associated are in non-coding regions of the genome. They're outside the genes, sometimes some distance away. It turns out that there are two explanations for that. As we've come with very large studies to find what we call the causative SNP, the one SNP that might be related in the region, those we commonly observe seem to be related to regulation of gene activity by affecting chromosomal contact or a transcriptional regulation. So they affect gene function as a regulatory mechanism, though they're not the actual coding gene. And that's why we use the term effector genes. They are responsible for the function of some direct effector gene.
So early on in the use of these approaches, your article mentions that they were associated with some problems with false discovery, with finding things that didn't actually turn out to be replicable. What was done to prevent that as the field advanced?
Well, when the field first started, very early, the first genome-wide association studies were done in about 2005. There's now been literally thousands and thousands and thousands of such studies. For example, in diabetes alone, there's been 370 plus studies in the GWAS catalog. The first tool, early on, we weren't sure what the level of significance would be. So what was done was replication. If you did a first study with 2,000 cases and 2,000 controls and found 10 significant SNPs, you went to another study and tried to replicate your findings and find that they find the similar results. That worked a little bit and was certainly anything that you confirmed was valid. But it turns out that you often missed in the second study because depending on your sample size, there's a variation in your ability to identify SNPs. So for example, the first GWAS for type 2 diabetes found a handful of SNPs. We're up to 1,289. But that's when we went from a couple thousand to 2.5 million. The initial GWAS for type 1 diabetes were about, again, a handful, 6 or 7. And now we're up to 81 in studies that involved 500,000 people. So one of the best assurances was to have more statistical rigor, meaning setting the threshold for acceptance higher, and larger and larger studies. Not always possible, but certainly for the major diseases, this was the way to go. I should emphasize here, and a very important aspect of genome-wide association studies, which we haven't commented on. It's very important that these be done in every ethnic group possible and in large sample sizes in each ethnic group. This is very important for both discovery, narrowing the regions that I just discussed, but it's also for importance for their use in a moment. And what we haven't discussed yet is the two major uses of this information. One to understand biology and one to use them for prediction.
In your article, you mentioned two uses of genome-wide association study results, one for predicting risk for individuals and one for understanding or elucidating mechanisms of disease. Can you speak briefly about an example in each of these areas that you think is particularly noteworthy?
Yes, let's talk about prediction first. We are actively using, I told you we would identify many SNPs. So now there's hundreds of SNPs for coronary disease, for breast cancer, and I told you there's about 80 for type 1 diabetes. Each of those three examples are actually now in the clinic. People are using polygenic risk scores for breast cancer to identify who should undergo not only more frequent mammography and at earlier ages, but also that should alternate with MRI of the breast. 2, for coronary artery disease, they're identifying people that are extremely high risk for which intervention should start at a young age, regardless even actually of lipid scores, because it turns out the genetic risks are somewhat independent of your lipid levels. And 3, for type 1 diabetes, these are being used in very intriguing studies to identify who's at risk in the population, who should undergo antibody screening, and who should therefore eventually be intervened to help delay or eventually even prevent type 1 diabetes.
And what about the use of GWAS study results for elucidating new disease pathways?
So I'll give you one or two examples. One is the very first one that we were involved in, Crohn's disease, published in Science in 2006, where we identified the interleukin 23 receptor that was involved in inflammatory bowel disease at all, and that's led to a whole development of biology based on that. A similar example in the lipid field is the GWAS done in 2010 by the Global Lipid Genetics Consortium, which found the SORT1 locus on chromosome 1, which was a key gene that's involved in lipid metabolism. And another insight, I'll give you just one more insight. It turns out that an important risk factor, of course, known well to this audience, is that coronary artery calcium is a major risk factor for coronary disease. What about the people who have no coronary calcium? What's their risk factors? Well, it turns out if you do a polygenic risk score for both coronary disease and for stroke, a polygenic risk factor for stroke is a risk factor for subsequent coronary disease events in those who are coronary artery calcium negative. So that tells you that the pathway in the people that are coronary artery calcium negative is somewhat different for coronary disease than those that are coronary artery calcium positive.
In your article, you specifically discussed the work by Hauser et al. Evaluating genetic risks for primary open-angle glaucoma. Can you talk a little bit about their work and what the results were?
The importance of that work was a genome-wide association done in African Americans. African Americans have the highest risk for glaucoma in the population. They get it earlier, they get it more severely, and they have more blindness. But until recent studies, such as those by Hauser and a more recent study in Nature Genetics, the studies in the African American population were not as available in the same sample size. By doing so, in the Hauser et al. Study, they identified a gene that interestingly provided a connection between Alzheimer's disease pathophysiology and glaucoma pathophysiology, which is the major biological insight from the Hauser et al. Paper published in JAMA at around the same time.
So, Jerry, this is obviously a complicated topic. What do you think are the key takeaways for our listeners?
The key takeaway is the Genome-Wide Association study and era is allowing us and has allowed us and is allowing us to take advantage of the fact that regions of the chromosomes and genes are inherited together in units in people and populations. This has allowed us to identify the variation in those genes that predispose to a whole host of different diseases. Now that we have identified those variations in those genes, they are used in two different ways. One is to create what are called polygenic risk scores or polygenic scores that are being increasingly used to predict disease in a variety of both clinical and public health models. And two, we've identified new causal pathways that were simply not recognized before. And these are allowing the development of new interventions to either treat or prevent these diseases.
I am Roger Lewis, and I've been speaking with Dr. Jerry Rotter about Genome-Wide Association Centers. You can find a link to the article in this episode's description. This episode was produced by Daniel Nassisi at the JAMA Network. To follow this and other JAMA Network podcasts, please visit us online at jamanetworkaudio.com or search for JAMA Network wherever you get your podcasts. Thanks for listening.